All right. Well, thank you everyone so much for joining us. This is our third live stream of the day during Ask a Scientist. My name is Katherine Hagman. I'm one of the organizers of the MSU Science Festival. Today, we're going to be joined by 10 different scientists from a variety of different scientific disciplines. And we are encouraging you to ask all of your questions. So come curious. Feel free to ask any of your questions in the comments section. We'll be sure to answer those for you. So next up, we have Dr. Joseph Hill, who is a postdoctoral fellow with the MSU Department of Horticulture. Thank you so much for joining us.
Yeah, glad to be here.
So to start, do you mind telling us a little bit about yourself and what your research is at MSU?
Yeah, so I've been at MSU for a few years, and our lab basically studies plant shape and how we can try to use that to improve agriculture. I could real quick show some pictures of what we have. Okay, so we basically study how a few different genes control the shape of trees. So here's an example of a couple of peach trees. On the left, we have a standard peach tree, and on the right, what we call a pillar peach tree. And you can see its branches just want to go just straight up. And similar, but opposite, we also have something called lazy, where this gene causes the branches to just want to go straight out. You can see on the left, that plant is really gnarly and outward, where again, we have a tack on the right that just kind of grows upward. So we're trying to understand how these genes work to control plant shape.
Great, wonderful. So can you tell me a little bit more about how you got to this point? So what did you study as an undergrad, and then what did your studies look like continuing? And what inspired you to take this path?
Yeah, sure. So I'm from the Carolinas. I went to Clemson University for undergraduate, and I studied biochemistry. I just kind of picked that major when I got to Clemson because just reading the description, it had to do a lot of the things that I liked in my high school biology class. Trying to understand how DNA and protein and those kind of things work on the level of the cell and smaller. So after I got my biochemistry degree from Clemson, I decided I really liked research and being in a lab. So the next step I thought was I'll get a postdoc, or sorry, no, a PhD. So I got into a PhD program at Penn State. And so for about 6 or 7 years, I was in Pennsylvania doing research on plant cell walls and how they make cellulose. So like cotton, what our shirt's made of, how plants are able to make it. And it's actually quite an interesting polymer because, wood and cellulose, we have paper, cotton, the timber that goes into our houses. It's a very versatile and strong material. And then I decided I wanted to do something a little bit different, a little bit more applied. So I came here to Michigan State, which has lots of great plant biology and horticulture. And now I'm, trying to do a little bit more research into something that might actually help, growers. So understanding how these plant shapes form, we can engineer or breed different kinds of trees, especially fruit trees, because, every year they just want to grow and grow and grow. And that's not so great for the growers because they have to prune and spend a lot of time and effort getting them in the right shape for, efficient harvest and efficient spraying and whatnot.
Great. So when you're working in the lab, what's your day-to-day look like? What's a typical day in your work?
Yeah, that's a great question. So I'm mostly a molecular biologist. So again, we're studying genes and proteins, and they're really, really tiny. So a lot of the times we're just moving around really small volumes of liquid. I actually have right here, this is what we call a pipette. So this is kind of how I interact with the world. It's just a small tool to suck up a very, very small amount of liquid. This is my largest pipette, and it can only take up 1 milliliter, which is a third of a teaspoon. And my smaller pipettes do about 1,000th of that. So we're moving small things around because we're trying to, understand how genes work and doing a lot of manipulations, designing genes or modifying them and seeing how that changes the ultimate shape of a plant when we put them back in a plant.
Mm-hmm. Can you talk a little bit more about that? How exactly do you genetically modify plants?
Yeah, so I guess real quick, I'll show our model plant. So this is Arabidopsis, and it's what we do a lot of our work in because it's very small and easy to use. It's kind of the plant world's version of the classic lab mouse. So it's very, very easy to genetically modify. We actually just make use of what nature already does. There's some bacteria called agrobacterium, and just in nature, they infect a plant, and they found a way to take a piece of DNA and put it in the plant. And that causes the plant cells to kind of grow out of control like a tumor and spit out a bunch of nutrients, which the bacteria then eat. So they're basically engineering their environment by changing the plant's genetics to better benefit them. And when researchers figured this out, 30, 40 years ago, they realized, well, we can move DNA around a little bit. We can just take out those nasty genes that cause the plants to grow out of control and put whatever we want in there. So, that when we're moving around little drops of liquid in lab, that's when we're shuffling the genes around, and then we put them in a bacteria, put that onto the plant, and let, essentially slightly modified nature just do its thing.
Mm-hmm. And so how does a GMO plant then differ from traditional plant breeding? What's the difference in those?
So like genetic modification versus traditional breeding. It's a great question. So it kind of depends on what kind of GMO you're doing, because you can do slightly different things. You can try to break a gene or add a new gene. But most of the time, what I think the good thing about genetic engineering is that you're making a small change, you know what you're doing, and in theory, you know what the outcome is going to be. But with breeding, it's a lot more effort and a lot more mixing things together. A metaphor or analogy I like to use is, in English, we have a lot of loan words from other languages. So let's just imagine we want to take a new word from Spanish and we start using that word a lot. It should probably go in the dictionary. So genetic engineering would just be taking that word and its definition and just adding it to the dictionary. One little change, put it in. Whereas breeding, well, you can't really do something so precise. You have to take an English dictionary and a Spanish dictionary and then take half of each one and mix them together. And then you try to make sure your new word's there. But now you have this horrible mixed up dictionary that lost half of your English words and has all these extra Spanish words. So to get it back, you have to again take half of that hybrid dictionary, mix it with an English dictionary again. And every time you want to make sure your one word is still there. But you have to keep doing this process over and over and over again. And eventually, in theory, you should be able to get back to an English dictionary with maybe like a page from the Spanish dictionary that has your word. So that's good, but it takes a lot of effort, a lot of time and resources. And you've actually brought along some extra words. So you get some unintended things as well. And when you do that first big mixing, you don't know what kind of things you're going to bring along. So in both cases, there can be unintended consequences. So it's just balancing that and trying to understand it. But that's why when we make new kinds of things, either through breeding or GMOs, you test them. You make sure that they're safe or delicious.
Mm-hmm. Yeah, that makes a lot of sense. You can get more specific with the changes that you're making.
That's the idea, yeah.
Could you give a couple examples of the types of plants that you work with and what sort of changes specifically that you're hoping to make right now?
Yeah, so again, the first one would be Arabidopsis. So this is kind of what a normal Arabidopsis looks like. You can see its branches go up like, most plants. And many times we're working with mutants. So you start out with a broken gene and you go, okay, this one gene is broken. Now what happens to the plant? And what does that mean for what the gene does? So this plant is what we call lazy. And you can see it's very different. The branches just kind of grow downward in this arc. And it turns out that lazy is involved in how the plant senses and responds to gravity. So we're trying to understand the different pathways involved in that, because we kind of know the end point and the beginning point. Just for those who don't know how gravity is sensed in plants, I have a little visual tool here. So organisms are made of cells, and in plants, a lot of the cells are like long rectangles. So we'll have this glass jar here as our example plant cell. And there's certain plant cells that are filled with these little balls of starch, and they're very heavy. So they fall to the bottom of the cell because the cell is basically just filled with liquid. So if the plant ever gets knocked over in some way, you can see the marbles I have in the bottom fall to the side. And just like in the plant cell, the little starch balls fall to the side, and the plant has sensors all around the inside of the cell, and it can sense, oh, look, we've changed direction. We should probably fix that. And lazy's somewhere after that, but we don't know all those steps in between. Hey, we're sensing a change, and now we're growing back upwards. So yeah, it's kind of like we know we're doing a road trip from New York to LA, but we don't really know any of the steps in between, and there's lots of paths you can take. So we're trying to identify some of those steps to better like figure out what the route is and understand how this whole process works. But we also work in apples, peaches, and plums. I showed, some pictures of peaches and plums before. We also work with apples, and like here's an example of a tiny little apple plant we have. So we keep them in these little boxes with what I call science jello, just full of nutrients and hormones, and grow them. Yeah.
Oh, that's great. Let's see, we've got just over 10 minutes here left. So for everyone tuning in and viewing, be sure to ask any of your questions for Joseph in the comments section, and I'll relay those over to him. Joseph, what are some topics that people can ask you about that you think might be relatable to their everyday lives?
So, of course, anything involving plants or food or genetics, things on the molecular level. So besides just plants, like I know things about viruses and heredity, human diseases, basically anything on that very, very tiny level. That's kind of my area of expertise, trying to understand how, you have something so small, smaller than a cell, a few things happen in there, and then you get a huge change, like, the shape of the entire plant changes from a few little changes at that very tiny level.
That leads me to another question. You are joining us for a couple other presentations during the Science Festival and this digital transition. Could you talk a little bit about what viewers can hope to hear in your next sessions?
Sure. I'm making a video, just general overview about kind of what viruses are, because there's a virus going around, if you hadn't heard. And people may be curious because the word gets thrown around a lot, but how do they work and what do they do? And are they that bad? Are they very common? And then I have one a little bit more about my work in understanding how plants and gravity function, which I kind of gave a little overview before with my visual aids.
Oh, we have our first question from a viewer. How long does it take from gene discovery to cultivar release for fruit trees, for example, apples or peaches?
That's a good question. It can take a really long time. It kind of depends on understanding exactly what the gene is doing. And you also have the problem that fruit trees just take a long time. If you're gonna do breeding that takes multiple generations, that's gonna take 20, 30 years potentially. It's kind of like the problem I was mentioning before with mixing the dictionaries. You might find one really good trait you like, and if you try to breed it in, maybe there's a disease resistance, but it's not a very good fruit. So you have to then breed that in to another plant with a good fruit, and then go through a few different generations to try to get both together in the same plant. So there's a little bit of luck involved. With genetic engineering, there's potentially a lot of regulation, though with new technologies like CRISPR, there's some ways to do a few limited things that aren't considered genetic modifications anymore. So that can actually be done relatively rapidly, maybe even 10 years, which is rapid for trees.
You mentioned CRISPR. For those of us who might
Yeah.
not know what that is, can you talk a little bit about that?
Sure. So yeah, that's probably been in the news a lot. And CRISPR is just a tool. So it's a molecular scissors. So if we have DNA, which controls a lot of our traits and genes, and CRISPR is just a really, really efficient way to hunt down and find a specific region of DNA or gene and cut it. And then again, we let nature do its thing. A lot of times once you make that cut, it doesn't fix it quite right. And that can, modify or disrupt a gene. And sometimes getting rid of a gene actually helps a lot. There's some genes that prevent a plant from defending itself from a pathogen. So we get rid of that gene and now they become more resilient to a pathogen because they can fight back because they're no longer like preventing themselves from doing that. And then you can do a bit more advanced things by just making the cut and then providing a new piece of gene to potentially fix a broken gene like we have in many human diseases. You have a problem with one of your genes, so you need a normal version inserted. And the reason it can sometimes not be considered genetically modified is once you get CRISPR in there, Cas9, and it cuts, then it's a completely natural process to repair that DNA and introduce an error. And as long as the CRISPR doesn't stay, it just goes away. There's no way to tell the difference between that kind of modification and something that happens just completely naturally, because mutations and these kind of changes are a very, very natural process. I think I've read, every new human has at least 60 or 80 mutations they're brand new to them that come from them dividing from a fertilized egg to a baby. And that's why even identical twins have a few differences, because after they split, they get a few mutations when they're dividing and becoming a whole baby.
Oh, that's fascinating. That's something new to me. Let's see. Well, we have about 10 minutes left, and we do have a couple other questions.
Sure.
Rita's wondering, do you ever use bacteriophages?
No, I never use bacteriophages for my work. In molecular biology in general, the bacteriophages were used a lot beforehand just to kind of manipulate and move DNA around. But nowadays we have lots of easier ways to just do that in a test tube. So we don't need to rely on those. They're kind of antiquated at this point. But, and I am going to mention this in one of my videos, but I guess I'll say it now. There are some countries that I know, maybe not now, but at least in the past, use bacteriophages instead of antibiotics, because for those of you who don't know, a bacteriophage is a virus that infects a bacteria. So you can use two different germs to basically fight each other. Use this bacteriophage to go kill some bad bacteria that's making you sick.
All right. Quite a few questions now. Let's see. Are there any ways to speed up the breeding process?
Yeah. One way I know that the USDA recently developed for, I think, mainly plum, is they inserted a gene that made the plants flower at a much younger age. So when you grow it from a seed, instead of taking 4, 5, 6 years before it can flower, and thus you can breed it, it can do that 1, 2 years. And then during the breeding, you breed out that gene. So it just allows you to get more breeding cycles in to do this, many steps of mixing I was mentioning. But in the end, you lose that genetically modified piece of DNA so that in the final product, it's not even there. So that's one good way. And recently in wheat, I know just through controlling like light, temperature, humidity, they've gotten it to where they can do, I think, 6 generations in 1 year. So every 2 months you can go from a seed of wheat to the next generation's seed of wheat without any kind of genetic or even hormonal changes, just light and temperature controls, very specific. Whereas before, I think you could only do, maybe 2 or 3 cycles a year. So that really is speeding things up.
All right, we have another question. Do you work with any poisonous plants?
Not really, though poison's all about dosage. So we do a little bit of work with tobacco, and nicotine is actually very poisonous. It's just how much does it take to be poisonous? So we usually consider something poisonous if there's only a little bit, but if you get anything in a large enough amount, it can be poisonous and kill you. But yeah, nothing that's like hideously toxic in the plant. Now, in the lab, there is some very dangerous chemicals. For example, this plant is actually one that I mutated with a chemical mutagen to go in and randomly disrupt the DNA. And as you might guess, randomly disrupting DNA is bad for everything that's alive. So that's a bit of a dangerous chemical.
Another question from Erin. She's wondering if seeds will still grow if they're not pointed straight up. So in this situation, she's wondering if there's a rock in front of the seed, would they still find their way, and how would they do that?
Yeah, that's a great question. That plays mostly into gravity. So when we think about plants, we think that they grow towards light because they use light and photosynthesis to make their food, their energy. But really, they actually use gravity probably even more than light. Because in the example you just gave, yeah, nothing is controlling the direction a seed goes down. I mean, perhaps a farmer could build a machine to do that, but for thousands and thousands of years in nature, there was, squirrels don't carefully plant the acorn in the right orientation. They just fall and get covered up. So underground, there's no light. The plants have to figure out, well, which way am I going to grow? And if they don't, they'll die. So they use this gravity signal to know, okay, I'm feeling gravity. This way is up. I'm going to grow up, with the idea being the sun's up. So if I just keep going up, eventually I'll get there. And if they'll hit an obstacle like a rock, of course they can't go straight through it. So as they push, they'll probably randomly go in one direction and then continue to find their way up. And then once they get into the light, that actually tells them, instead of growing straight up, to spread out, because you want to get a lot of leaves and capture as much sunlight as you can. So the plants aren't really growing toward the light. Instead, what happens is when they get shaded, like, let's see, here you go. My arm, there's a bit more light on this side's a bit shaded. So on this side of my arm, if it were a plant stem, there would be no light. So in the absence of light, it would go, oh, we should grow a little bit. And it'd start to bend the plant. And you see that gets it more towards the light. So it's really, instead of growing towards the light, they're growing away from the shade. And gravity is kind of this universal thing always there, telling them which way is up, because they're kind of like fighting. Gravity's saying go straight up, but when they hit a light, they're going, we need to go out. We need to capture all this light. And then, there's differences between different plants. So trees, they go straight up and they grow really tall, but bushes tend to just grow out.
We have 5 minutes left. So again, if anyone has any questions, go ahead and enter those into the comments section. We do have a full day full of live streams until 4:30, so be sure to stay tuned. Joseph, thank you again for joining us today. This is wonderful. Really looking forward to your future talks in the coming weeks. Yeah. Is there anything else you want to share with everyone before we sign off here?
Yeah, I guess I have something here I could share. So one type of thing we do in lab a lot is use something we call a reporter. So we're studying a gene, we want to know how it works. So if you want to know how anything works, you have your who, why, what, when, where, those kind of questions. So a reporter is something to tell you where. So it's some type of signal you can see, whether it be a color or light, to know like, okay, well, in this part of the plant or this part of the animal, that gene is on. So we have a few different reporters that actually glow or light up. This is one of our Arabidopsis plants with firefly luciferase in it. So it's literally the gene in the bud of a firefly that makes it light up. You put it in a plant, and what's happening is whatever signal we're looking for is, you get that light lighting up the plant. But of course, it's very dim, so you have to use a long exposure camera. So we've stolen that gene from firefly because we do that kind of thing a lot, especially in molecular type biology. We just find something in nature and go, hey, that's a great idea. We're just going to use that. Just another example, here's a white light image of a plant on its side, and you can see when we put it in the dark, you can see the luciferase lighting up more in the bottom of the stem because it's trying to reorient itself to gravity. And then the other kind of light or glowing plant we have is stealing a gene from jellyfish. So if you guys have ever heard of bioluminescence, a lot of deep sea creatures make their own light or fluorescence. And this is a cucumber with an engineered bacteria that glows on it. So this is white light. If we turn it in dark or in the fluorescence, we can see in this illuminated part, that's where that pathogen, that bacteria, is infecting the cucumber. And this type of glowing isn't, it's not like the firefly where you can just see it. It's like a black light. If you've ever gone to, like cosmic golf or bowling or something where they have black lights and paints on the wall glow up, it's that kind of thing where if you put a specific light on it, it'll glow up really brightly. And yeah, again, that's something we stole from jellyfish.
Very cool. So why? What leads to this type of research? And
well, yeah, it's like I was saying, it's trying to answer those questions about, if we want to figure out how something works, answering when and where it's being expressed or where that gene is, or that protein. It can be really informative just to be able to see, especially on this tiny level. It's not like studying birds or deer where you can go out and map where the populations are and how they're moving. In a cell, we can see a cell with a microscope, but the stuff smaller than a cell, we can't really see unless we tag them. If we attach one of these glowing things to them, then we can watch that glow move around, and it can be really informative to go, oh, look, it's starting in this part of the cell and then moving over here and then moving here, and that corresponds to this other thing that's happening. So yeah, it can just being able to see can tell you a lot. And it's one of the things we take for granted in the macro world that we don't have in the molecular world unless we do, a weird trick like this.
we have 1 minute left, and we just got a fantastic question.
Ooh,
we got into this
good.
earlier. Someone is asking, what would happen if a seed is planted in space without planetary gravity?
Yeah, so that's actually basically what lazy is here. So lazy can't really sense gravity, so it just kind of goes out and all over the place and down. So they've done experiments where they grow plants in space, or if you take a plant in regular gravity and just constantly rotate it so that it can never sit still and feel gravity in one particular direction, it mimics being in microgravity or no gravity. And they grow a whole lot like this, where they just kind of go out all crazy in no particular direction. And one of the things I'm actually studying, I mentioned that this plant was actually a mutant. It's actually a double mutant. It has 2 mutations. It started out as one of these lazy guys. And then I've made a mutation in some other unknown gene that has somehow fixed that. So that's one of my projects right now, is trying to identify what gene I changed to fix that. And I would imagine, and it'd be a cool thing to try in the future, is to send that into space. Because if it fixed lazy in gravity, and that's inability to sense gravity, maybe it'll do the same in space. Maybe the idea is that now it's growing a lot more due to light sensing rather than gravity, is one hypothesis.
God, that's incredible.
Yeah, it's fun. Yeah.
All right. Well, we're at the end of our session. Thank you again for joining us. This was really
Yeah,
fantastic.
it was a
Again,
pleasure. Thanks,
forward
guys.
to your sessions next week.
Yep.
All right.